next speaker, Dr. Javier Márquez-Ruiz from Seville, Spain. Javier.
Okay, thanks. Thank you for the invitation to participate in this great and amazing meeting. I'm really happy to be here. So the idea is to, I would like to focus on some considerations about how can we translate all this information we take from the animal, and what are the main problems we have when we try to do that. So please, next slide. Okay, so this is the idea. The idea is how can we replicate this in the small animals. In this case, we want to know what happened in the brain of small rodents, because that means that we can combine with transgenic animal, for example, in order to explore some pathological conditions. So maybe one of the big problem is what about the brain size and the geometry. This is how the human brain looks like in comparison with mouse. You can see that we have a huge difference in not only in the brain of on the side of the brain, but also in the geometry of the brain. So if you can see here, in the case of mouse, there is no convolution. So that means that we are going to obtain some, in some aspect, we are going to obtain some simplified result from TDCS. This is good because we can try to validate different models in this simple cortical geometry. But that means also that we need to take care when we want to translate to the humans TDCS. So please, next slide. The other point is that most of the papers in human used to compare before versus after TDCS. But when we try to explore about the mechanism in animals, sometimes we focus in the effect during the current. So that means that we normally think that the plastic effect is going to move in the same direction that we observed during the stimulation. But as you can see here, this is not always true. For example, if we explore sensory potentials, we obtain no after effect, even when we observe a beautiful during effect for anodal currents. Okay, so that's one point. Next, please. The other important point that I also have mentioned here before is what about the brain state of the animal. Of course, we need to consider not only in vivo versus in vitro, but also we need to take care when we compare anesthetized result in comparison with alert. Even during the alertness, you have some fluctuations in brain state that is going to induce many important changes. So that's the reason why some previous papers have published that there is no effect with TACS, for example, in the awake animal, but they observe something in the anesthetized one. So next slide, please. So the second point is not only about the animal model, but also when we want to record single neuron activity, there are also many parameters that we need to consider. One of them is, for example, what about the recording place? Sometimes we record far away from the active electrode, so that means that probably the intensity is not the maximum and the effect is not going to be optimized. The other important point is that for that reason, we need to identify the real electric field. So that's something that we can do in animals, because we can record this experimental electric field, and this is important because we need to validate the different diffusion models. And also, in our experience, we have huge change depending on the morphology and the orientation of the cell. So that means that even when you are in a region, in a very similar region from one from one recording to the other, the effect is going to change a lot depending on the orientation. In this particular case, in this picture, you can observe the opposite effect depending on the orientation of a Purkinje cell in the cerebellum of an awake animal. So finally, I would also mention that in the last few years, we have more and more papers that focus on TACS. I think that we have also a nice result in our lab, but I think this is because we used to have more success when we try to record neuronal changes during alternating current. So I would like to mention that probably for TDCS, we need longer experiments, more similar to human, that imply, for example, 10, 20 minutes of neuronal recording. That I know this is something that it is really difficult to do in animals, but probably this is something that we need to do as soon as possible in order to make the interpretation of TDCS more than TACS. Of course, TACS is also very interesting. So that's all by my side. Thank you for the invitation and for this nice meeting.
Thank you very much. Thank you very much, Javier.